Hello, today we are starting chapter 2 to talk about proteins, which is a biological molecule. Okay, here are your Cambridge objectives. We're going to talk about protein structure and function as related to biology, enzymes, antibodies, and muscle contraction. And talk about the structure of an amino acid and the formation and breakage of a peptide bond. So today we are going to do a dissection. But you don't need your scalpel because we're not doing a typical animal dissection. Instead, we'll be dissecting a bacterial cell. So we just looked at bacterial cells in the last few lectures. We can look through our microscope to find a rod-shaped cell. Now we can imagine that we could pick up the cell, slice it open, and see inside. We would look closely and you would start to see some major cell structures like a flagella or a cell wall, and maybe even some ribosomes. But if you zoom in a little bit more, you start to see individual proteins floating around inside the cell, catalyzing chemical reactions. So many of the components of a cell are made up of macromolecules, which simply means large molecules formed by linking together small molecules. The major molecules, macromolecules, within the cell include proteins, nucleic acids like DNA and RNA, carbohydrates, and lipids. In this lesson, we will discuss the structure of these macromolecules and break them down into their individual units, all the way down to individual atoms. So to keep track of the elemental building blocks of our macromolecules, I want you to take notes and remember that in this lecture we're talking about proteins, but the next few videos, which are going to be lipids and carbohydrates, are connected to this one. It's lipids, carbohydrates, and proteins all together as our macromolecules section. Proteins play important roles within a cell. Some make up the structure of cell components like a flagella or a pilus, and many play the role of enzymes, which catalyze reactions necessary for life. They're an extremely important class of macromolecule in all living organisms. More than 50% of the dry mass of most cells is protein, and they have many, many important functions. So let's take a look at some proteins. So as you can see here in this picture, they are complicated structures. And they can be made up of hundreds to thousands of individual units called amino acids, which are all linked up in a chain and then folded like origami into complicated shapes. So we learned in our last unit that these proteins are put together amino acid by ribosomes. Okay? So all enzymes themselves are proteins. Proteins are essential components of cell membranes. Some hormones are even proteins. For example, insulin and glucagon are. The oxygen-carrying pigments, hemoglobin and myoglobin, are proteins. Antibodies, which attack and destroy invading microorganisms, are proteins. Collagen, which is another protein, adds strength to many animal tissues like bone and the walls of arteries. Hair, nails, and the surface layers of skin contain the protein keratin. Actin and myosin are the proteins responsible for all muscle contraction. And proteins may be storage products, for example, casein in milk, and ovalbumin in egg whites. And this is just a tiny amount of the examples of protein that make up our bodies or important components inside of our bodies. So the structure is complex, but all proteins are actually only made up of about 21 different amino acids, just in many, many different combinations. Every amino acid has the basic structure shown here, consisting of carbon, hydrogen, oxygen, and nitrogen, and this would be called the backbone of the amino acid. They all have a central carbon atom, which is bonded to an amine group, which is NH2, and a carboxylic acid group, which is COOH. It is these two groups which give amino acids their names. So the third component that is always bonded to the carbon atom is usually a hydrogen atom. The only way in which amino acids differ from each other, in fact, is the remaining fourth group of atoms bonded to the central carbon. Okay, and this is a different molecular group that hangs off to one side. Most of the special side groups, which are called R groups, contain carbon, hydrogen, oxygen, and nitrogen. But there are 2 amino acids that contain sulfur. So there are a total of 20 different amino acids which occur in the proteins of living organisms and all have a different R group. So take a look here. You can see that of these 20 amino acids, they're all the nitrogen, hydrogen, carbon, and oxygen. And then the difference between each one is that R group. So if you look down, sorry, let me get my pen. If you look down, here's sulfur and sulfur in this one. Okay. Okay, so let's talk about the bonds between amino acids. These are called peptide bonds. Okay, so one loses a hydroxyl, which you guys will come to remember is an OH group, from its carboxylic acid group, while the other loses a hydrogen atom from its amine group. So this leaves a carbon atom of the first amino acid free to bond with the nitrogen atom of the second. So you guys should remember this from STEM. We learned about condensation reactions. Okay, so you can see here, here's an amino acid here, and a second amino acid here. They're coming together to bond. Okay, and so in the process of doing this, they lose a water. Okay, and so then as they bond, the carbon is free, since a water molecule was lost, the carbon is free to bond with the N. Okay, and so here is the peptide bond. The new molecule, which has been formed, made up of 2 linked amino acids, is now called a dipeptide. Any number of extra amino acids could be added to the chain in a series of condensation reactions. So you have a condensation reaction that occurs here, you lose a water molecule, there's the bond between the carbon and the nitrogen, and this could keep on going and going and going, building a long protein. A molecule made up of many amino acids, so 2 amino acids is a dipeptide, but one made of many is called a polypeptide. And it's another example of a polymer and a macromolecule, which is similar to, or it's like a polysaccharide. A complete protein molecule may contain just one polypeptide chain, or it can have 2 or more chains which interact with each other. Okay, so now we're going to talk about the different structures. We're going to talk about primary, secondary, and tertiary quaternary structures of proteins, along with the types of bondings that we see there. Okay, so in general, when we're talking about protein structure, you can think about 4 different types of structure that they might have, or you can also think of it as phases in a structure. So there's a primary structure. The primary structure of a protein is a sequence of amino acids linked together by peptide bonds, forming a polypeptide. Okay, and then next you see the secondary structure. Local regions of the resulting polypeptide can then be coiled into a helix, okay, and here it's an alpha helix, and we're going to learn about what that means, to form a secondary structure. So you can see the primary structure is just the linked amino acids into polypeptides. Then those polypeptide chains coil into a secondary structure. And then for the tertiary structure, regions of secondary structures associated with each other in a specific manner form tertiary structure, which describes the final folding of the polypeptide. So you can see in this really good picture that you have, here are the coils of the secondary structure, okay? So they're all in this structure, and this coil is sort of bending on itself and wrapping around itself. And then lastly, you have the quaternary structure. So for multimeric proteins, the quaternary structure describes the association of 2 or more polypeptides as they interact to form the final functional protein. So you have a tertiary structure, and here you can see there's 4 tertiary structures that are combined into one final quaternary structure. Okay, so let's dive into those a little bit. Okay, so the primary structure, a polypeptide or protein molecule may contain several hundred amino acids linked into a long chain. The particular amino acids contained in the chain and the sequence in which they are joined is called the primary structure of the protein. So that's really important to understand. Okay, the sequence in which they are joined and The specific amino acids that are in the chain, that's what makes up the primary structure. So we're not talking about how it's coiled or how it's folded or how it fits with others. Just what particular amino acids are in there and what order they're in. Okay, so that's primary structure. Primary structure is a linear chain of amino acids. So again, what amino acids are in there and what order are they in? There are an enormous number of different possible primary structures based on the order of amino acids. Even a change in one of them can completely alter the properties of the polypeptide or protein. Okay, secondary structure. So regions stabilized by hydrogen bonds between atoms of the polypeptide backbone are how the secondary structures are formed. So the two main types of secondary structures are alpha helix, that's what that symbol A, okay, that italicized A is alpha helix, or beta-pleated sheet. Okay, so you can see an example there of an alpha helix, which is like a coil, or the beta sheet, which looks like sort of like a folded piece of tinfoil or piece of paper. Okay, so let's talk about each of those. So let's first focus on alpha helix. A polypeptide chain, or part of it, will coil into a corkscrew shape called an alpha helix. And it is a secondary structure due to hydrogen bonding between the oxygen of the CO group of one amino acid and the hydrogen of the NH group of the amino acid 4 places ahead of it. So if you look at that picture, you can see that the polypeptide is the long chain, right, of amino acids in the sequence that they're in. But then when they start to coil, those elements are coming close together. And as we learned when we talked about water and how important hydrogen bonds are weak bonds that occur between elements or molecules that have partial charges. So you can see that hydrogen and that oxygen, okay, are polar, they're creating polar molecules, and so hydrogen bonds are forming in between those elements. And so what that's doing is that's making the coil a little stronger, and it's helping it to keep its shape, even though we do need to remember that hydrogen bonds are not strong bonds. Okay, so now we'll talk about the beta-pleated sheet. Sometimes hydrogen bonding can result in a much looser, straighter shape than the alpha helix, which is then called a beta-pleated sheet. Hydrogen bonds, although strong enough to hold the alpha helix and beta-pleated sheet structures, are easily broken by high temperatures and pH changes. So you can see the way that these are bonding, or where the hydrogen bonds are occurring, are different than in the alpha helix. Some proteins or parts of proteins show no regular arrangement at all. It all depends on which R groups are present, and therefore what attractions are occurring between amino acids in the chain. So in diagrams of protein structure, A helices can be represented as coils or cylinders, and beta sheets as arrows, and random coils as ribbons. Okay, tertiary structure. So in many proteins, the secondary structure itself is then coiled or folded. So tertiary structure is the compact structure of a protein molecule, resulting from the three-dimensional coiling of the already folded chain of amino acids. So remember, we have the primary chain, which is just what amino acids are there and what sequence they're in. And then we have the secondary, which is the either coiling or the pleating of those, depending on the R groups and where hydrogen bonds are formed. And then again, so they're already coiled, and then again they're folding. Okay, and so that secondary folding is called tertiary structures. And then in a quaternary structure, this involves the clustering of several individual peptide or protein chains into a final specific shape. A variety of bonding interactions, including hydrogen bonding, and then some different ones, ionic bonds, which are called salt bridges, and disulfide bonds hold the various chains into a particular geometry. And again, whether it's a hydrogen bond, an ionic bond, or a disulfide bond depends on the R group and the shape of the individual structure. There are two major categories of proteins with quaternary structures, fibrous and globular. So there are four main types of bonds that we just mentioned to help keep folded proteins in their precise shape. So first you have hydrogen bonds, and these can form between a wide variety of R groups. Then you have disulfide bonds. These form between two cysteine molecules, which contain sulfur atoms. Then you have ionic bonds, which form between R groups containing amine and carboxyl groups. And then you have hydrophobic interactions, which occur between R groups which are nonpolar or hydrophobic. Okay, so talking about that salt bridge. Salt bridges, or ion bonds, are a special form of particularly strong hydrogen bonds made up of the interaction between two charged residues. A disulfide bond is a single covalent bond between the sulfur atoms to two amino acids called cysteine. So disulfide bonds form between different amino acids of a protein chain and then help to stabilize it and maintain a distinct three-dimensional form. They are really important in determining the tertiary structure of the proteins, and they're also important in determining the quaternary structure of some proteins. Okay, so just some more information. I encourage you to pause the video at any of these models and really take a look at these structures. It's really important that you understand where these bonds are occurring and that you're able to see it in the models that I'm providing. Okay, fibrous proteins. Fibrous proteins are insoluble in water. Okay, ones like keratins in wool and hair are composed of coiled alpha helical protein chains with other various coils, analogous to those found in a rope. So you can see there to your right. So other keratins are found in skin, fur, hair, wool, claws, etc. Actin and myosin in muscle tissues, and fibrinogen needed for blood clots. So you can see you have an alpha helix, which is then coiled into two alpha helices, which are then, those pairs are coiled, and then you have a filament where there's four right-hand twisted filaments. Globular proteins. So these are proteins whose molecules curl up into a ball shape. And it's easier to remember these when you hear the names. It's the, looking back at the fibrous proteins, right? These look like fibers of what fiber of hair or wool would look like. And then globular proteins are, they're in like a ball shape. Okay, so they're somewhat soluble in water and often have a spherical or roundish shape. Many globular proteins have roles in metabolic functions. Their precise shape is the key to their functioning. Like enzymes, for example, are globular proteins, and we know that their precise shape is key to their functioning. Okay, so here is a table of the characteristics of fibrous versus globular proteins, which you do need to remember.
So, in a living organism, proteins may be found in cells and in other aqueous environments such as blood, tissue, fluid, and in the phloem of plants. Globular proteins usually curl up so that their nonpolar hydrophobic R groups point into the center of the molecule, away from their watery surroundings. So water molecules are excluded from the center of the folded protein molecule, and the polar hydrophilic R groups remain on the outside of the molecule. And therefore, they're usually soluble because water molecules cluster around their outward-pointing hydrophilic R groups. So here you can see the hydrophilic exterior, which you can see is on the outside of the globular protein, and then the hydrophobic interior are in the interior of the protein. Okay, so hemoglobin is an example of a globular protein, and it is the oxygen-carrying pigment found in red blood cells. It is made up of 4 polypeptide chains, so it has a quaternary structure, and each chain is itself a protein known as globin. So as you see here in this model, you see 1 chain, 2 chains, 3 chains, 4 chains. Each chain is a globin, and it's the 4 together in its quaternary structure, which equal a hemoglobin molecule. So there are many types of globin. 2 types are used to make hemoglobin, and these are known as alpha globin and beta globin. 2 of the hemoglobin chains, called alpha chains, are made from A globin, and the other 2 chains, called B chains or beta chains, are made from beta globin. And I really need you to go in your textbook, not right now, but after the video, and read page 43 about sickle cell anemia. So you can do that in class or on your own. Okay, heme groups. So each polypeptide chain of hemoglobin contains a heme group. A group like this, which is an important permanent part of a protein molecule, but is not made of amino acids. It's called a prosthetic group. Okay, let's look at one. So each heme group contains an iron atom. One oxygen molecule, O2, can bind with each iron atom. So a complete hemoglobin molecule with 4 heme groups can carry 4 oxygen molecules. So that's 8 oxygen atoms at a time. And it's this heme group which is responsible for the color of hemoglobin. This color changes depending on whether or not the iron atoms are combined with oxygen. If they are, the molecule is known as oxyhemoglobin, and it is bright red. If not, the color is purplish. Okay, collagen is a really good example of a fibrous protein. Collagen is the most common protein found in animals, makes up 25% of the total protein. It is insoluble fibrous protein found in skin, so skin, tendons, cartilage, bones, teeth, walls of blood vessels, even more. It's really an important structural protein, but not just in humans, in almost all animals, and is found in structures ranging from the body walls of sea anemones to the egg cases of dogfish. A collagen molecule consists of 3 polypeptide chains, each in the shape of a helix. So this itself is not a helix, it's not as tightly wound. So let me show you. Okay, so each complete 3-stranded molecule of a collagen interacts with other collagen molecules running parallel to it. Covalent bonds form between the R groups of amino acids lying next to each other, which you can see. These are covalent bonds between R groups. These cross-links hold many collagen molecules side by side, and which together form fibrils. And the ends of the parallel molecules are staggered. If they were not, there would be a weak spot running right across the collagen fibril. So finally, many fibrils lie alongside each other, forming strong bundles called fibers. And I really like these 2 pictures from a scanning electron micrograph because each one you can see something a little different. So in the left one, the one with the red fibrils, you can see the triple helices lying parallel with one another. And then here, on the right, you can see all of the fibers that are lying side by side. Okay, that's it. Thank you for watching. The next 2 will be lipids and carbohydrates.